हाय एवरीवन, वेलकम बैक टू बर्मा स्टडी। तो एक बार फिर से आप लोगों का एनसीईआरटी लाइन बाय लाइन रीडिंग सीरीज में स्वागत है। आज हम लोग प्लांट किंगडम का कंप्लीट एनसीईआरटी रीडिंग करने वाले हैं। तो चलिए बिना किसी देरी के वीडियो को स्टार्ट करते हैं। मेक श्योर आप लोगों ने चैनल को सब्सक्राइब कर लिया होगा। Chapter 3 Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whitaker 1969, wherein the suggested the five kingdom classification: Monera, Protista, Fungi, Animalia and Plantae. In this chapter, we will deal in detail with further classification with kingdom Plantae, popularly known as the plant kingdom. We must stress here that your understanding of the plant kingdom has changed over time. Fungi and members of the Monera and Protista having cell wall have now been excluded from Plantae through earlier classification, placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophyte, pteridophyte, gymnosperm, angiosperm under Plantae. Let us also look at classification within angiosperm to understand some of the concerns that influenced the classification system. The earliest system of classification used only gross superficial morphological character such as habit, color, number and shape of leaf etc. They were based mainly on vegetative characters or on the androsium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to the vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment as against the natural classification system developed which were based on natural affinities among the organisms and consider not only the external features but also internal features like ultrastructure, anatomy, embryology and phytochemistry. Such a classification for flowering plant was given by George Bentham and Joseph Dalton Euchre. At present, phylogenetic relationship system based on evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. This becomes more important when there is no supporting fossils evidence. Numerical taxonomy, which is now easily carried out using computer, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior and chemotaxonomy, that uses the chemical constituent for the plant to resolve confusions, are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll bearing simple thalloid, autotrophic and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitat, moist stone, soil and wood. Some of they also occur in association with fungi, lichen and animals. Example, sloth bear. The form and size of algae is highly variable, ranging of colonial forms like Olvex and the filamentous form like Eulothrix and Spirogyra. Figure 3.1. A few of the marine forms such as kelps from massive plant bodies. The algae reproduce by vegetative, asexual and sexual method. Vegetative reproduction is by fragmentation. Each fragment develop into a thallus. Asexual reproduction is by the production of different type of spore, the most common being the zoospores. They are flagellated, motile, and one germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix, and non-flagellated, non-motile, but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in a species of Eudorina is termed as the anisogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed as oogamous. Example: Volvox and Fucus. This is the diagram of Volvox and Eulothrix. Porifera and Polysiphonia. Now, algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producer of energy-rich compounds which form the basis of the food cycle of all aquatic animals. Many species of Porifera, Laminaria and Sargassum are among the 20 species of marine algae used as food. Certain marine brown and red algae produce large amount of hydrocolloids, water holding substance. Example: algin. Brown algae and Carizen red algae, which are used commercially, agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparation of ice cream and jelly. Cholera, a unicellular alga rich in proteins, is used as a food supplement even by a space traveler. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae and Rhodophyceae. Now let us discuss about Chlorophyceae. Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, filamentous. They are usually grass green due to the dominance of pigment chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein beside a starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made up of an inner shell of cellulose and outer layer of pectose. Vegetative reproduction usually takes place by fragmentation. Asexual reproduction is by flagellated zoospore produced in the zoosprongia. The sexual reproduction show considerable variation in the type and formation of sex cell, and it may be isogamous, anisogamous or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Spirogyra and Chara. Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous form ectocarpus to profusely branched form as represented by calves, which may reach a height of 100 meters. They possess chlorophyll A and C, carotenoids and xanthophyll. They vary in color from olive green to various shades of brown depending upon the amount of the xanthophyll pigment. Fucoxanthin present in them food is stored as complex carbohydrate which may be in the form of laminarin or mannitol. The vegetative cell have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastid, a centrally located vacuole and nucleus. The plant body is usually attached to the substrate by holdfast and has a stalk, the stipe and leaf-like photosynthetic organ the front. Vegetative reproduction takes place by fragmentation. Asexual reproduction in moist brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogamous species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common forms are Ectocarpus, Dioptiota, Laminaria, Sargassum, and Fucus. Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment or phycoerythrin in their body. Majority of the red algae are marine with greater concentration found in warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depth in ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as fluoride starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spore and sexually by non-motile. Table 3.1 Division of algae and their main characters. Classes. Common name: major pigment, stored full, cell wall, flagella or habitat. Chlorophyceae, Phaeophyceae or Rhodophyceae. Chlorophyceae common name is green algae. Major pigment: chlorophyll A and B. Stored food: starch, cell wall, cellulose, flagella, 2-8 equal apical. Habitat: freshwater, brackish water, salt water. Phaeophyceae common name is brown algae. Major pigment is chlorophyll A, C and fucoxanthin. Stored food is mannitol, laminaria. Cell wall is cellulose and algin. Flagella is two unequal lateral. Habitat is freshwater, brackish water and salt water. Rhodophyceae common name is red algae. Major pigment is chlorophyll A, D, phycoerythrin. Store food is fluoride starch. Cell wall is cellulose, pectin and polysulfate sulfators. Flagella इसमें absent होता है. Habitat is fresh water, brackish water, salt water most. Gamete sexual reproduction is oogamous and accompanied by complex post-fertilization development. The common members are Polysiphonia, Poriphyra, Gracilaria and Gelidium. Now we move to bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded area in the hills. Figure 3.2. Female thallus and male thallus. Funaria and sphagnum. Bryophytes. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid and shaded localities. They play an important role in plant succession on bare rock soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true root, a stem or leaf. They may possess root-like, leaf-like or stem-like structure. The main plant body of the bryophyte is haploid. It produces gamete, hence is called a gametophyte. The sex organ in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellate antherozoites. The female sex organ called archegonium is flask-shaped and produces a single egg. The antherozoites are released into water where they come in contact with archegonium. An antherozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. A sporophyte is not free living but attached to the photosynthetic gametophyte and derive nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spore. These spores germinate to produce gametophyte. Bryophyte in general are of little economic importance, but some mosses provide food for herbaceous mammal. Birds and other animal species of Sphagnum moss provide peat that have long been used as a fuel and as packing material for transshipment of living material because of their capacity to hold water. Mosses along the lichens are the first organism to colonize rock and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into leaveroids and mosses. Now, liveroids. Liveroids grow usually in the moist, sandy habitat such as bank of streams, marshy ground, damp soil, bark of tree, and deep in the woods. The plant body of a liveroid in thalloid, example Markentia. The thallus is dorsiventral and closely approached to the substrate. The leafy member have teeny leaf-like appendage in two rows on the stem-like structure. Asexual reproduction in liveroid takes place by fragmentation of thalli or by the formation of a specialized structure called gamete. Singular gamma. Gamete are green multicellular asexual bud which develop in a small receptacles called gamma cups located on thalli. The gamete become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organ are produced either on the same or on different thalli. The sporophyte is differentiated into a foot, seta, and capsule. After meiosis, a spore are produced within the capsule. These spores germinate to form free living gametophyte. Now, let us discuss mosses. Mosses. The predominant stage of the life cycle of moss is the gametophyte, which consists of two stages. The first stage is the protonema, a stage which develops directly from a spore. It is creeping, green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axis bearing spirally arranged leaf. They are attached to the soil through multicellular and branched rhizoids. This stage bear the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organ Antheridia and Archegonia are produced at the apex of the leafy shoot. After fertilization, the zygote develops into a sporophyte consisting of a foot, seta, and capsule. The sporophyte in mosses is more elaborate than that in liveroids. The capsule contains a spore. A spore are formed after meiosis. The mosses have an elaborate mechanism of a spore dispersal. Common example of mosses are Funaria, Polytrichum, and Sphagnum. Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purpose and as soil binders. They are also frequently grown as ornamental. Evolutionarily, they are the first terrestrial plants to possess vascular tissue, xylem and phloem. You shall study more about this tissue in chapter 6. The pteridophytes are found in cool, damp, sandy places, though some may flourish while in sandy soil condition. You may recall that in bryophyte, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte which is differentiated into true root, a stem and leaf. These organs possess well differentiated vascular tissue. The leaf in Pteridophyta are small microphytes as in Selaginella or large macrophytes as in fern. The sporophytes bear sporongia that are subtended by leaf-like appendage called sporophytes. In some cases, sporophytes may form distinct compact structure called strobili or cones. Selaginella equisetum. The sporongia produce a spore by meiosis in a spore mother cell. The spore germinate to give rise to inconspicuous, a small but multicellular free-living, mostly photosynthetic thalloid gametophyte called prothallus. These gametophytes require cool, damp, sandy places to grow. Because of this specific restricted requirement and the need for the water for fertilization, the spread of living pteridophyte is limited and restricted to narrow geographical region. The gametophyte bear male and female sex organ called antheridia and archegonia. Respectively, water is required for transfer of antherizoids and the male gamete released from the antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular well differentiated sporophyte which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia which produce two kinds of spore, macro and micro. spore are known as heterosporous. The megaspore and microspore germinate and give rise to female and male gametophyte respectively. The female gametophyte in these plants are retained when the parent sporophytes for variable periods. The development of the zygotes into young embryos takes place within the field gametophyte. This event is a precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Coelopsida, Lycopsida, Lycopodium, Sphenopsida, Pteropsida, and Pteris. 3.4 Gymnosperm. The gymnosperm are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post fertilization are not covered, that is, are naked. Gymnosperm include medium-sized tree or tall tree and shrubs. One of the gymnosperm, the giant redwood tree Cecropia, is one of the tallest tree species. The roots are generally taproots. Roots in some genera have fungal association in the form of mycorrhiza pinus, while in some other cycas small specialized roots called coralloid roots are associated with nitrogen-fixing cyanobacteria. The stems are unbranched or branched. The leaves may be simple or compound. In cycas, the pinnate leaf persists for a few years. The leaves in gymnosperm are well adapted to withstand extreme of temperature, humidity, and wind. In conifer, the needle-like leaf reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperm are heterosporous. They produce haploid microspore and megaspore. The two kinds of spore are produced within sporangia that are borne one sporophyll, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspore develop into a male gametophyte generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grain takes place within the microsporangia. The cones bearing megasporophyll with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be borne on the same tree Pinus. However, in the cycas, male cones and megasporophylls are borne on different trees. The megaspore mother cell is differentiated from one of the cell of the nucleus. The nucleus is protected by envelopes and the composite structure is called an ovule. The ovules are borne one megasporophyll which may be clustered to form the female cone. The megaspore mother cell divided meiotically to form four megaspore. One of the megaspore enclosed within the megasporangium develop into a multicellular female gametophyte that bears two or more archegonia or female sex organ. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes in gymnosperm, the male and the female gametophytes do not have an independent free living existence. They remain within the sporangia. Retained on the sporophyte, the pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tube carrying the male gametes grow towards archegonia in the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, a zygote develops into an embryo and the ovules into seed. These seeds are not covered. 3.5 Angiosperm. Unlike the gymnosperm, where the ovules are naked, in the angiosperm of flowering plants, the pollen grain and ovules are developed in a specialized structure called flower. In angiosperm, the seeds are enclosed in fruits. The angiosperm are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest olivia to tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicine, and several other commercial important products. They are divided into two classes, the dicotyledons and the monocotyledons. Dicotyledon and monocotyledon. Summary: Plant kingdom include algae, bryophyte, pteridophyte, gymnosperm, and angiosperm. Algae are chlorophyll-bearing, simple, thalloid, autotrophic, and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into three classes, namely chlorophyceae, phaeophyceae, and rhodophyceae. Algae usually reproduce vegetative by fragmentation या sexually by formation of different type of spore and sexually by formation of gamete which may show isogamy, anisogamy or oogamy. Bryophytes are plants which can live in soil but are dependent one than that of algae. It is thallus-like and prostrate or erect and attached to the substrate by rhizoid. They possess like leaf-like and a stem-like structure. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thyloid and dorsiventral, whereas mosses have upright, slender axis bearing a spirally arranged leaf. The main plant body of a bryophyte is gamete producing and is called a gametophyte. It bears the male sex organ called antheridia and female sex organ called archegonia. The male and female gamete produce fuse to form zygote which produce a multicellular body called a sporophyte. It produces haploid spore. This spore germinate to form gametophyte. In pteridophyte, the main plant is a sporophyte which is differentiated into true root, a stem, and leaf. These organs possess well differentiated vascular tissue. The sporophyte bearing a sporangia which produce a spore. The spores germinate to form gametophyte which require cool, damp places to grow. The gametophyte bear male and female sex organ called antheridia and archegonia respectively. Water is required for transfer of male gametes to archegonium where zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperm are the plants in which ovules are not enclosed by any every wall. After fertilization, the seed remains exposed and therefore these plants are called naked-seeded plants. The gymnosperm produce microspore and megaspore which are produced in microsporangia and megasporangia borne on the sporophyles. The sporophylls, macrosporophylls and megasporophylls are arranged spirally one axis to form male and female cones respectively. The pollen grain germinates and pollen tube release the male gametes into the ovule where it fuses with the egg cell in archegonia. Following fertilization, the zygote develop into embryo and the ovule into seed. The angiosperm are divided into two classes, the dicotyledons and monocotyledons.